So on behalf of the Biochemical Society and the Portland Press, I'm really delighted to welcome you today to this webinar, which is part of the Biochemistry Focus series. And this is a series that covers research areas in the molecular biosciences, as well as practical sessions to support career development. And the Biochemical Society welcomes suggestions for future topics and speakers in the webinar series. So you can see on the Biochemical Society website for more details of how to propose a topic or a speaker. So I'm Dr. Claire Friel. I'm a protein biochemist from the University of Nottingham. And it's my really great pleasure today to introduce Professor Dan Rigden, who's from the Department of Biochemistry and Systems Biology at the University of Liverpool. And so Dan today is going to give us some insights into AlphaFold 3, which I'm sure you're aware is a state-of-the-art tool for modeling protein structures and protein complexes. And it can also model bound ligands, post-translational modifications, and nucleic acids. And results from AlphaFold can complement or can even substitute experimental structures. And its power is increasing rapidly with every new edition of AlphaFold that comes out. And so we're now on AlphaFold 3. However, it's very important to understand the quality of the models that are produced and to understand the limitations, to have an appreciation of the limitations of these models. And so that's what Dan's going to help us with today. And his seminar is going to cover how to obtain models in AlphaFold 3 and the strengths and weaknesses of these models, and also go over a selection of the many uses of these models. So Dan is going to field questions after the presentation, but you'll be able to submit your questions during the presentation by typing them in the question box. And we'll try to answer as many of those questions as the time allows. So I'll now hand over to Dan. I'm really excited to hear his presentation.
We'll get going. Well, thanks for the introduction. It's nice to be here and have a chance to tell you about AlphaFold 3. So what we're going to talk about is some of the basics, a little bit of the comparison, a little bit of the history, what AlphaFold 3 gives you, which is not just coordinates, it gives you quality measures as well, and you should know what they are. We'll talk about how to get AlphaFold 3 models. Then we'll spend quite a bit of time talking about its performance in different situations. And at the end, a little bit more detail on some of the alternatives to AlphaFold that have come along that have some particular unique selling points. And I should say, just before we get going, a couple of warnings. I'll need to go relatively quickly over these things, but we have plenty of time for questions at the end, so you can, if I've skated over anything too quickly, you can bring me up on it and we'll talk into it in more detail. And also, I'm going to be showing you a lot of very recent data from preprints, very largely. So, of course, there is the possibility that after peer review, the picture will change slightly, but these things are changing so quickly that to see the state of the art, you really need to be looking at the preprints rather than just the papers. So just a bit of history. I mean, AlphaFold 3 is obviously the latest in the lineage. AlphaFold unnamed or unnumbered AlphaFold came about in 2018. This was already very exciting, was by far the best method at the time, but it was essentially the pinnacle of the previous kind of idea about modeling, which is to predict distograms here, distances between residues in one step, and then in a second step transform that information into a 3D structure. This came to prominence at CASP 13. It's worth just making sure everyone knows what CASP is, because it comes throughout the talk. So CASP is an experiment every 2 years where people give groups who develop the software unknown structures, and they will try and predict those structures, and then we test how well the different methods compare to each other. So CASP 13 was in 2018, and AlphaFold was already great at that time, quite exciting. But of course, much more exciting was 2 years later, when this two-step process was replaced by a single step. AlphaFold 2 produced these amazing structures, many times as good as an experimental structure. And indeed, 2 of the 3 Nobel Prize winners last year received the award for their work on AlphaFold 2. What we're talking about today, of course, is AlphaFold 3. So this is just explaining some of the key differences between AlphaFold 2 and AlphaFold 3. I'm imagining that most people on the line have used AlphaFold 2 and have some familiarity with it. The main difference is here. So instead of a protein-specific structure prediction module in AlphaFold 2, we have a general module that can work with different kinds of molecules, and that allows modeling of different complexes, nucleic acids, ligands, whatever you like. The inputs are still the same, so it can still use templates. So if your sequence matches something in the PDB, that information can be factored in. If you want it to, there may be reasons why you don't, but it can factor in. And then very important, where there isn't a template, AlphaFold 3 will only work well if it's got a good multiple sequence alignment. And by good, I mean lots of sequences with a sufficient diversity, and that allows it to read out what's called covariance, evolutionary covariance information. And in the absence of a template, that covariance information gives it the starting point to produce an accurate model. So as you'll see in a couple of different places, that's very important. The end point is, it gives you the same three outputs as AlphaFold 2. So the coordinates are what everyone's excited by, but it's very important to judge those coordinates in the context of the quality prediction information that it also gives. It gives us local and global estimates of model quality. And so you should not just look at the coordinates, you should look at the coordinates and assess their scores of different kinds. And this is just a reminder of those scores. So the PLDDT is a measure for each residue of how confident AlphaFold is that the environment of that residue is indeed correct. And so we've all become familiar with this kind of color coding, whereby you want to see lots of dark blue because that very confident structure. So typically familiar domains like these immunoglobulin domains are very confidently modeled, therefore they're dark blue. At the other end of the spectrum, we have the orange or red, which often has this kind of extended spaghetti-like structure, and that's often corresponds to intrinsically disordered regions. So AlphaFold will always give you a structure for the protein, but it's saying I have no confidence in this. And indeed, this would be a part of the protein that doesn't have a fixed structure, which is continually jiggling around in solution. Just in passing, it's interesting to note that one limitation of AlphaFold is highlighted by the structure on the right. These domains, these confident domains, are actually extracellular, and here we have a transmembrane helix. So all of this red spaghetti should be inside the cell on the other side of a membrane, but AlphaFold has no idea that membrane is there. And so it's giving us back a nonsensical structure where the spaghetti is wound around the extracellular part of the protein. The third output it gives you that you need to look at is the PAE, the predicted aligned error. And this is more useful for looking at the confidence in the relative orientation of different domains within the same protein. And so if you consider this protein here, we've got 4 reasonably confident domains, but each of them is connected by a piece of flexible protein structure. This is sometimes called beads on a string. And so what you see in this situation in your PAE, each of those domains manifests as a dark green square here. The dark green means that the distance between residue 10 and residue 20 is confidently predicted. And so if you've got a block like this, that corresponds to a folded domain in which all of those distances are reliable. The power of the PAE is when we look off the diagonal. So here's the first domain, here's the second domain, and we can ask, is the orientation of the first domain with respect to the second domain confident? And AlphaFold is saying, absolutely not. I've got no idea if this is right. And that makes intuitive sense, of course, because we've got these linkers between the domains. These domains will be jiggling around, sampling all kinds of relative orientations. And therefore, we have low confidence that this particular single snapshot has any special meaning. So why are we excited about AlphaFold 3? Well, these are the results from their paper last year. And the key innovation is being able to model heterogeneous and diverse biomolecular systems. So here they are showing results for different kinds of challenges where we're trying to dock ligands in this first panel here, and we're comparing to a standard conventional docking program called AutoDock-Vena and doing a little bit better. We're modeling nucleic acids here, and I'll show you more detail on this as we go through. We're modeling nucleic acids and doing better than other methods, but not quite as good as human-based groups. We're modeling covalent modifications, glycosylations, things reasonably well. And in terms of the comparison with AlphaFold 2, we're modeling single proteins a little bit better, protein complexes a fair bit better, and hard protein complexes, and we'll talk about why they're hard later on, much, much better. So plenty of good reasons to be excited about AlphaFold 3. Of course, it still has limitations, and one limitation is, I mentioned before, if there's no template in the PDB, there's nothing similar it can match your sequence to, then it is reliant on a multiple sequence alignment to extract that key covariance information to give it an initial idea of how the protein folds up. And so for a very small number of proteins, but if you're extremely lucky, you might have, unlucky, you might have one of these, you would do a blast of the databases, not find many homologs. Your MSA is quite shallow, and therefore the models that you try to make will be very bad. Here's a protein in the PDB, but it's a protein that these methods still can't model well. This is the AlphaFold database, AlphaFold 2, another version of AlphaFold 2, even AlphaFold 3. It can't model it well because there isn't a high quality MSA to do the modeling from. So that's a slight negative for a very small number of proteins, but the positive here is just very recently this whole new protein sequence database is coming along, which is vastly bigger than these already huge databases that have been previously used. So if you use AlphaFold 2, AlphaFold 3, you'll be looking at this kind of database here, which has a couple of billion sequences. That's a lot, but sometimes it's not enough. This new database, Logan, has 100 billion sequences, absolutely astonishing. And so this will make a big difference to those hard cases. We will be able to make much better models. And they actually show that in the preprint here. They take 50 difficult virus proteins. Virus proteins are harder because they evolve quicker, and therefore it's harder to collect the homologs in a nice MSA. And they show that the predicted quality of the models that you make when you make an MSA with Logan is much better than when you make an MSA with standard methods down here. So all of these proteins which are producing pretty horrible, useless models are suddenly producing high quality models with the benefit of this massive new database. So that's a real positive outlook. Some little characteristics of AlphaFold 3 that's a little bit different to AlphaFold 2 in some cases that you should be aware of. So we talked about AlphaFold 2 and what I call this red spaghetti, this kind of disorder, and how it represents as these kind of long extended squiggly regions. In AlphaFold 3, for some proteins, disorder actually appears as these helices. So this immediately looks more visually appealing. You say, oh, that looks like a nice model. This doesn't look like a nice model. But these helices are totally fantastical. Then they don't exist. If you predict these regions with conventional tools, you realize they are disordered and they shouldn't be folding up into something that looks ordered, like all of this helix here. So the AlphaFold 3 people who are well aware of this when they're developing it, and they even tried to train AlphaFold 3 to produce results that looked a bit more like AlphaFold 2. As they said, give us our red spaghetti back, but they didn't manage it entirely. So you may get results that look like this. But the key point is that you can see those helices are very low confidence. So you just need to be aware that big chunks of low confidence helix are likely disorder in the same way that the red spaghetti we're used to seeing is also disorder. So like any of these deep learning methods, you sometimes get clashes where atoms have come too close together. They There are clashes that are even worse than this, but these are some relatively modest classes where the side chains are just put so close that PyMol is thinking they're bonded. Of course, they're not bonded to each other, but the deep learning method doesn't have a great idea of the physics of these systems, and so you will get these bumps. For some purposes, that doesn't matter. If you're just looking at the overall fold, that's okay. But if you were to do some dynamics on this, of course, it would explode. So you need to be aware of that. One interesting quirk of AlphaFold 3 compared to AlphaFold 2, because it can model heterogeneous systems with other ligands present, you can sometimes get better models by completing the system in a sense. So this shows an example of that. This is some protein structure work from this guy. He was talking about it on Blue Sky. They had cryo-EM structure for this domain here, and they were trying to complete it by using AlphaFold 3 to predict the transmembrane region at the top here. And initially, they were getting terrible results. They had different results between human and mouse, which of course makes no sense. And the results were all quite low quality. And what they said, then they had their brainwave of saying, okay, we know this region is in the lipid bilayer. Maybe we can mimic the bilayer by adding in some fatty acids. So you can perhaps see on the right-hand side, this simulation here, they've added in fatty acids, which indeed make two kind of layers on the protein. And all of a sudden, the protein is feeling it's in its normal environment, and AlphaFold 3 rewards you with models that are much more confident and, of course, consistent between the human and mouse. So they call this the fatty acid hack, but this is actually much more general. So if you're modeling a calcium-binding protein, you want to stick a calcium in there. It will immediately make the model much more confident. Okay, so how do you get AlphaFold 3 models? Well, the easy way is to take advantage of Google DeepMind's generous server here, alphafoldserver.com. You can run 30 jobs a day. These jobs can be very big. So 5,000 tokens. A token here means either a residue or a base. So 5,000 residues is an absolutely huge system. So this will likely cope with even large systems that you're interested in. A token is an atom for other kind of ligands, so you use up your tokens a bit more quickly, but nevertheless, most of your needs, typical needs, will be catered for here. So you paste in your sequences, you say this is protein, this is DNA. It's got a relatively limited range of ligands on the server, so AlphaFold 3 can work with any ligand, but the ligands available in this library are a bit limited, but you've got your standard things like ATP, and you've got a bunch of metals like magnesium here. So that's very easy. But it's also relatively straightforward if you're relatively computer literate to install it locally. You don't need a massive machine, you just need a machine with a powerful GPU like this one. But it has to be admitted that it's not so convenient. So the input here, when you run it locally, is not just a command line with a FASTA file. You need to assemble a JSON file, which is more of a machine readable format and a bit of a pain in the neck, really. Having said that, just last week, there's this tool which I haven't tried called JAG maybe, which will help you assemble that JSON. So things will get easier, and you shouldn't rule out the possibility of installing it and running it locally. And then we'll talk about some of these in more detail later on, but just to say at this point, AlphaFold 3 has spawned a whole lot of lookalikes. Some of them are kind of inspired by AlphaFold 3, some of them try to replicate the AlphaFold 3 approach exactly. They have different groups behind them, some industry, some academics, some a mixture. It's quite driven by the relatively restrictive license in AlphaFold 3 in terms of not allowing commercial use, but it's also driven by people who just want to understand, want to explore the detailed operation in a bit more detail than AlphaFold 3 allows. In broad terms, I think you'll see as we're going through, there's nothing that, in a lot of basic tasks, there's nothing that's better than AlphaFold 3, but often these methods are almost as good and may in some cases be better, as I'll show you. If you want to try these methods, there are a couple of quite useful websites. These are commercial. I've got no connection to them, obviously, but they give you a certain amount of free access. And they've each got massive panels of these deep learning tools with a nice interface that you can just run a certain number of jobs per week or per month for free. And so it's a good way of trying some of these alternatives. And if you want to try AlphaFold 3, bolts and chai, I'm going to shamelessly plug our own software written partly by a PhD student in my group, Luke Elliott, which makes it very easy to run all three. So if you've got AlphaFold 3, but you don't have bolts and chai installed, it will install them as the job proceeds. It will do all the conversion of the inputs. It will prepare and allow you to compare all of the different models against each other, visualize them, show the clashes. I talked about the clashes before, where you can see the clashes and count the clashes, see which model has the fewest clashes and so on. It does genuinely make running these methods locally quite a bit easier. And the one important point I want to address here, which I think is kind of underappreciated. So bolts and chai, they are intended to replicate exactly AlphaFold 3, but we find repeatedly that one of them will just by chance perform better than another on a particular protein. So here we're showing exactly that. Here's a protein. There's nothing special about this protein, but you can see in this particular case, chai is giving us much more confident models than AlphaFold 3 and bolts. We don't know why, but if you really want to go the extra mile on a protein of interest and compare the three, you might find that one method outperforms the other, and that's good to know. So moving on to AlphaFold 3 and sort of various tasks you might want to challenge it with. You might want to be modeling a single protein chain. We could already do that very well with AlphaFold 2, but AlphaFold 3 is a little bit better. And that's also the message from the latest CASP exercise, where the groups that used AlphaFold 3 or both did statistically better than the groups that just use AlphaFold 2. So it is definitely a little bit better, at least. There are little tricks that some of these groups use to go beyond what your vanilla AlphaFold 3 could do in terms of accuracy. So one group realized that removing the intrinsically disordered regions improve the models a little bit. So you want the absolute best model, or you've got an edge case where things are not quite falling into place easily, you might try tricks like that. More challenging, perhaps more exciting, is AlphaFold 3 to predict protein-protein interactions. And so you can imagine two different scenarios here. Sometimes you know that two proteins interact and you just want to understand how. Sometimes you may suspect that two proteins interact, or you may have some other data that suggests some kind of connection between them, and you want to ask AlphaFold, is that connection between them because they do actually interact in a way that I really didn't suspect before. You could do this already with AlphaFold 2, but AlphaFold 3 is distinctly better. And then we'll come on to this. This works particularly well for cases where the two proteins have kind of co-evolved and where there's evolutionary pressure to maintain that interface. So if there's evolutionary pressure on the interface, the function of proteins A and B is to interact with each other, then you can extract that evolutionary covariance information that we talked about before. You can extract intermolecular evolutionary covariance, which is very useful for driving the modeling to the right answer. And then I'll mention a bit later, the built-in scores for AlphaFold 3 are not actually ideal for hard cases of protein-protein interaction. So there are some new scores that have come along. So this we saw before. This just says again, AlphaFold 3 is a little bit better than AlphaFold 2. But again, we've seen that at the most recent CASP. So at CASP, they ran an AlphaFold 2 baseline, which performed quite well, but it's not the best. AlphaFold 3 here performed a bit better, and you can see certain groups managed to outperform the vanilla AlphaFold 3 again with a variety of little tricks. These are probably not necessary in most cases, but it's worth knowing that you can do better if you try harder than you might get with the AlphaFold 3 server. If you're in a position of wanting to say, do these proteins interact? You know, I've got a bunch of proteins in the same cellular compartment. Do those proteins interact with each other? So questions like that. Then a really great piece of software is called Alpha Pull Down. And I like it because the name actually gives you an idea as to how it works. So you're familiar with pull downs in the lab where you immobilize an antibody on a column or something, and then you see what binds to it. This is doing the same in silico. So you have a bait protein here, and then you ask Alpha Pull Down to model that bait protein with a variety of different other proteins. You can do all against all, of course, but pull down mode is one against several. And it will efficiently do all of that modeling, give you a variety of scores. And sometimes those scores will tell you that, yes, your bait protein does interact with that protein, because the structure that you're getting is so confident and reliable that even in the absence of any experimental information, you can be confident those two proteins interact. So Alpha Pull Down makes these screens very easy. And here's an example recently where people were looking in a database. These are genetic codependencies. So this is a way of linking together two genes or proteins. And one reason they may be linked is because they physically interact. And that's indeed what they found for this protein here, which binds to a variety of other peroxin proteins involved in peroxisomal import. So there's lots of these exercises now. You can go fishing and say what protein interactions are present in a particular compartment or whatever. And you can even extend that idea to a whole proteome. You can say, I've got an organism, I've got all of these proteins in this organism. I'm going to sample pairwise all of those different combinations and see what interacts with what. And of course, even for a small genome like this mycobacterium, there's an awful lot of pairwise interactions to explore here. But these guys had a very clever strategy where they were modeling 10 proteins in a go, but they were simply ensuring that each pair was represented at least once in one of these runs. So AlphaFold 3 doesn't mind that you've got 8 of the proteins here that are completely random, not interacting with anything. It will still show you that these two proteins are interacting. And so in this way, you can sample cleverly all of these different pairwise interactions in only 2,000 or so jobs. I say only, that's still a lot. And kind of amazingly, these people did it on the AlphaFold server that I talked about before. So they just had regular personal accounts, 30 jobs a day, but there was a bunch of different people. And within 68 days of hammering the server with 30 jobs a day each, they accumulated this data set. So that's kind of inventive and impressive. And this just shows how all of the interactions in this known complex here were indeed discovered as pairs in these big kind of simulations with 10 proteins. So the green and the cyan here, they truly interact. They were present as a pair with 8 other random grey proteins, which are irrelevant, but the scores still allowed us to pick out that interaction. Okay, so I mentioned that some situations are harder because we don't have the evolutionary covariance. Those proteins have not co-evolved to maintain that interaction. One situation in that category is antibody-antigen modeling, and this work here shows that AlphaFold 3 is clearly better than AlphaFold 2 and these two competitors for antibody-antigen. But they showed this, this is a sentence from the preprint. This is a message that you're going to see repeatedly in the rest of the talk. The performance declines for complexes lacking structural similarities to the training set. So this is the idea that these deep learning methods are great, provided they've seen something a little bit similar before. If you challenge them with something totally new, they perform a lot less well. It doesn't mean they don't work at all, it just means it works a lot less well. And you'll see this message several times. So if you've got antibody antigen, you want to try AlphaFold 3. Here is another situation where there isn't that co-evolution. So this is a host pathogen situation. These are phage proteins binding to bacterial and human proteins. This is just to show you that although it's much harder when there's no covariance, it still does work. There are many phage bacterial protein interactions that they discovered by modeling them pairwise and spotting when they've got a really good, reliable complex. I mentioned that there's new scores come along for assessing these complexes. These address a number of issues. One is shown here. So IPTM is the built-in score from AlphaFold 3. It's from 0 to 1, 1 being the best. And in an ideal world, you would just simply look at this and you say, this score is close to 1. This must be a good interaction, a confident interaction. The problem is that the score is influenced by the amount of disorder in your proteins. So here are two proteins interacting. This is a genuine interaction, very nice high score, everything looks great. You had the same proteins with disorder tail on one or both of them, all of a sudden this IPTM is decreasing. And the more disorder you have, the lower the IPTM, even though the actual interaction between those two globular domains is exactly the same. So you're getting a falsely pessimistic idea of the score when you've got too much disorder there. So there are new scores, the active PTM and the IPSI score, which are both inspired to try and do a bit better. Here is the active PTM, which works particularly well on these protein peptide interactions. You can see when you add these extensions, when you add the disorder, the conventional score goes down, the active PTM goes down much less. And this IPSI score also has been independently validated on design proteins. So this is the current, this week's verdict on what's the best score, but it may be overtaken by events, of course. Just talking about protein peptide interactions, here are two benchmarks. Again, the simple message really is that AlphaFold 3 is better than these alternatives. But interestingly, both of these benchmarks commented that AlphaFold 3 suffers from structural memorization. So as soon as you go outside the training set, the performance decreases. And exactly the same message over here. But essentially a nice, clear, simple, consistent view from these two independent exercises that AlphaFold 3 is the best, actually. So AlphaFold 3 and nucleic acids. This is new over AlphaFold 2, of course. Nucleic acids are much harder to fold up because the covariance is less abundant and less useful. But nevertheless, basically AlphaFold 3 does almost as good as any server. So there are specialized servers for folding RNA. AlphaFold 3, which of course does many other things, is almost as good as those specialized servers, but you still need human input for the absolute best performance. Another paper here, they were looking at AlphaFold 3 modeling of DNA hairpins. Two interesting things here. Again, this message that where this structure is quite novel compared to what is seen before, then the performance is indeed reduced. But the second message here, which again you'll see repeatedly, is that deep learning and physics can complement each other, and you can get the best performance by a pipeline that includes them both. So here, for example, the initial model from AlphaFold 3, they ran some molecular dynamics. So that's introducing an understanding of the physics of the system. And in many cases, that's improving the quality of the model. So this combination is still a very current theme. Let's see, I'm running a bit short of time. I will try and accelerate. AlphaFold 3 and drug binding or ligand binding. So here there's good news and slightly less good news. The good news is that for modeling a single ligand into a protein, you can often get an extremely accurate result, better than you would with traditional tools. But again, the performance depends on whether it's seen something similar before, and also, less conveniently, there is no built-in score which you can read out as directly correlating well with the experimental binding affinity of that ligand. So here, for example, we've got the binding affinity on the x-axis. This is one of the scores they looked at. It kind of correlates moderately well, but you get the same score for 3 ligands with very different actual binding affinities. So it's no use for large-scale drug screening, for example. And another issue that you will find with AlphaFold 3 and all deep learning models, to be fair, is around the stereochemistry and the chirality of small molecules. So you'll find other papers that talk about how this can go wrong, but we've been exploring it ourselves. This is another PhD student, Luthfi, who's been looking at glycans. This is AlphaFold 3, and this is bolts, a new version of bolts that's supposed to do better with chirality. Indeed, it does. But I'm really just showing you here some of the grotesque errors that you can find in the outputs of these methods. So AlphaFold 3 in these first 3 columns, you get these extra bonds from the clashes. You also get, these are hydroxyl groups on sugars, which have suddenly become double bonds. This is a pyranose ring, which has suddenly become planar and so distorted that it shows as aromatic in pymol. Horrible things can happen, and it's really quite surprising because your overall message is these are miraculously effective tools, but certainly sometimes you can get really eye-openingly bad results for certain ligands. But just to say that this is AlphaFold 3, purple is bad here. Bolts 1, where they introduce some extra restraints on chirality, does much better, but you do still get these errors even with bolts 1x. So just be very careful in the ligands. Always eyeball them, I would suggest. Covalent inhibitors are much better. There was a preprint just about them, a relatively small bit of work, but naturally, if you've got a covalent bond between your inhibitor and the protein, that restrains the conformational possibilities a lot. And they also found that this built-in score was very useful for ranking. So covalent inhibitors are a bit of a special case. They are much better, but we still have some kind of stereochemical issues with them. AlphaFold 3 can model post-translational modifications. In this work, they were looking at PTMs versus drug binding, and nothing works terribly well. There was again some cases that work better with one tool than another. That message as I gave you before, that if one tool doesn't work, try another. It may work because there are differences between these tools. But here's an example of a kind of clear explanation for why phosphorylation of this residue changes the binding of this ligand. So it does work in some cases, but the other point, of course, we've talked about training. There are many fewer structures with PTMs, post-translational modifications, in the PDB than there are proteins in general. So it just has less information to work with. So I think we can expect it to work less well for the moment. Just a bit about AlphaFold 3 and similar tools versus structural biology. So for a long time, people have been using AlphaFold models routinely for molecular replacement. That's all perfectly standard. The only couple of things I wanted to say here are that AlphaFold 3, the PODDTs, these quality measurements, they are now per atom. So each individual atom will get a PODDT rather than a residue. And this does actually improve the performance of these models in molecular replacement. So there's a clear reason to consider using AlphaFold 3 instead of AlphaFold 2 for molecular replacement. And in particular cases, which we've been monitoring how many cases can be solved molecular replacement with these models, the answer is almost all of them. But there are occasionally examples where AlphaFold 3 can solve something and AlphaFold 2 can't. So here the AlphaFold 3 model allows for a better splitting of the individual domains with our CCP4 slice and dice software, and that allows you to solve the structure with an AlphaFold 3 model, whereas AlphaFold 2 over here wouldn't solve it. These methods are also having a big impact on cryo-EM. So you can do basic things like make a model with AlphaFold 3 and fit it into your cryo-EM map. Sometimes you want to do something a bit more sophisticated. So it's known, for example, that different methods can produce different states of the same protein. Here is a transporter with an inward and an outward state. And sometimes the modeling method will just prefer one when you would quite like the other one because that's what you've seen in your map. So cryobolts, for example, is taking your map and using it to push the modeling. This is the bolts modeling occurring, and in two different steps it uses features from the map to influence the modeling and push it in the right direction, so that when you've got your final model, it fits very well into your map. You can also use the kind of ligand binding options of AlphaFold 3 and similar tools to provide initial estimates of where the ligand is binding. So this was work using Chai, which is another, I haven't mentioned it yet, but here you've got a kind of typical blob where the ligand is, but it's not unambiguous by any means. You don't know exactly where the ligand fits based on that map feature. What you can do is ask Chai or another tool to predict where the ligand's going to be, fit it in, and then you can do some refinement with molecular dynamics or similar. So again, what we're seeing here is the effectiveness of combining the deep learning method to place the ligand and then the physics-based method to refine that placement, which, you know, improves its fit. Okay, so a couple of slides just on particular unique selling points of, what am I doing for time? Almost there, yes, I can see. Particular new selling points of some of the rivals of AlphaFold 3. So one of them is called Proteinics. Two things to mention about Proteinics. There's a Proteinics mini version, which is just astonishingly fast. We've got an application where we want to generate very large numbers of models. And we've realized that with the right kind of configuration, a model up to 200 residues takes 1 second. It's almost inconceivable on a perfectly normal cluster. It's amazingly fast. We also find that Proteinics, this is work from another student, Marga, we also find that it's more reliable at modeling covalent modifications. Here, the covalent modification is the tetrahedral intermediate of hydrolase enzyme. So we can model it. You will notice again these slightly wonky angles. So it's not perfect, but it is very convenient and works more reliably than AlphaFold 3 in our hands. Bolts 2 works quite well, maybe not as good as AlphaFold 3, but it has one really valuable feature, which they're justifiably excited by. In drug design, you want to have reliable ways of predicting the affinity between a ligand and your protein. You can do that in quick and dirty ways that don't work very well, or you can do these kind of gold standard ways that are extremely slow. They've devised a deep learning method which works very well, almost as good as these gold standards, but much, much quicker. So if you're interested in protein ligand affinity, Bolts 2 is the way to go. And then just to mention another exercise with a cool name, Boltzina. This is a combination of Boltz and Autodoc Vena. And what they do here is use the fast docking of Vena, combine it with the excellent affinity estimates of Boltz, hence Boltzina, and try and get the best of both worlds. And then I know I'm pretty much out of time, but you may be thinking AlphaFold 3 sounds great. Is AlphaFold 2 obsolete? The answer is definitely not. Several reasons. One is AlphaFold 2 is the basis of the AlphaFold database. And the AlphaFold database has a lot of interesting new features that you may not have seen if you haven't been there recently. So it has these alpha missense predictions of pathogenicity. So in other words, the likely detrimental consequence or otherwise of a mutation at a given position in the protein. And it will also split the AlphaFold models up into domains and classify those domains within the CATH database, which is also very cool. There are lots of AlphaFold 2 based databases of proteins, mainly protein-protein interactions, but also databases that fill in the gap of virus proteins, which are not in the AlphaFold database. And another thing in favor of AlphaFold 2 is it's better understood, and there's a much more mature kind of ecosystem of methods around it. One particular sub-area of methods is all to do with conformational sampling, and people have done endless different ways of manipulating with the MSA to give you slightly different structures out of AlphaFold. And I'm just highlighting this one, AF2 to RAVE, AF2 RAVE, because again, it combines the benefits of deep learning and physics. So you get models out of AlphaFold 2, you run some MD, and you get enhanced conformational diversity from the integration of the deep learning and physics methods. So just some kind of obvious conclusions. AlphaFold 3 is fantastic, but you still need to interpret the results with some care. There will be some trickier situations. As you've seen many times, it will struggle, or at least perform less well, when you go beyond its training data. And people are well aware of this. So the openbind.uk initiative, for example, is about producing protein ligand complexes on a really large scale to help train the next generation of these deep learning methods. For the moment, a real trend is combining deep learning and physics-based methods, and you've seen that many times. And the other impact of AlphaFold 2 and 3 is really just inspiring people to look at deep learning applications for all sorts of other purposes. So there's the one I picked out as a really high-profile example here is this bioemu, which is a really fast deep learning shortcut to get conformational sampling. So MD without the time-consuming calculations. So yeah, I will stop there. I think I've gone slightly over. Apologies for that, and I'll take any questions.
Hi, and we've got quite a few questions here. I'll apologise now if I mispronounce anybody's name, but I'll do my best. So we have a question from Alula Robert, who asks if you have any thoughts on what is the most informative experimental approach to validate AlphaFold structures?
Oh, that's a good question. I suppose it depends what you're reading out of the AlphaFold structures. I mean, if it's just a new protein fold, then any structural biology method will be equally good to confirm that. If you're looking at conformational sampling with AlphaFold, then the kind of gold standard that people use along MD trajectories, and you sort of you test whether you're exploring the same modes of structural variation as you see in the MD. Yeah, I guess it depends is the answer to that.
Yeah. Dorothea Gerkovich is asking if there's an upper limit in AlphaFold 3 for testing how multiple proteins interact with one another. I think that means an upper limit in number of proteins. I'm interpreting that as.
Well, if it's the kind of upper size limit of a complex, then I mean, like I say, for the AlphaFold server, it's 5,000 residues. If you go locally, then it will just simply depend on the GPU memory, and well, for 5,000 is already, that's going to ask for a lot of your GPU. So there are methods that sort of assemble things piecemeal. So people have done that in the past, things like the nuclear pore complex, which are absolutely vast. They've kind of asked the question, okay, how does protein A bind to protein B, and then separately, how does protein A bind to protein C, and then you can kind of put together the jigsaw from the subcomplexes. Yeah.
And there's another question that was somewhat related to that. I can find it now. I'll just ask the questions in order because I can't find the question I was thinking of. Samantha Gibbons asks, are you seeing that it's becoming more common for biochemists to use alpha pull down to complement pull down experiments from the lab, or perhaps even to completely replace wet lab experiments?
Yeah, good question. I haven't seen people use it to confirm an actual, I mean, I just haven't seen that. I mean, in general, that kind of alpha pull down fishing expedition is becoming very common. Lots of people doing that. But yes, I mean, it is true that the ideal is to combine that with experimental work. I mean, you know, I say that the scores will sometimes show you unambiguously that the two proteins bind to each other, but yes, if you can go and confirm that in the lab, so much the better. Yeah, and likewise, I guess if you've done a pull down in the lab and you want to say, do these proteins interact directly, or is there possibly a third protein interacting in the middle, then alpha pull down will help you answer those questions. Yeah.
Tobias Fabluck asks, when modeling protein-protein interactions, how do the PAE scores of AlphaFold 2 and AlphaFold 3 compare? And he says he's asking because he sees in most cases that the scores are very different between AlphaFold 2 and AlphaFold 3.
Well, I mean, I think if AlphaFold 2 and AlphaFold 3 have both got the right answer, then the PAEs, those figures should look quite similar, really. I think if you had to say, no, that's wrong, and in fact they differ, which do you believe? I mean, you would intrinsically go for AlphaFold 3. It is generally better, you know, and it is better at detecting those protein-protein interactions. So probably would also be reflected in being better in the scores that it attaches to those protein-protein interactions as well.
So this is the question I was looking at for before. Hannes Giovanazzi is asking, how does AlphaFold 3 deal with large multimeric membrane protein complexes such as photosystem 2? Do you have any knowledge of that?
Well, I mean, basically, there's no innate problem of membrane proteins. You just have to be aware that AlphaFold 3 has no concept of the membrane being there. And it actually can be a kind of useful filter. We've seen this ourselves when we're sometimes is that, you know, if you've got two proteins that you believe, you're wondering if they interact, they've got some kind of borderline score perhaps, you can ask, you know, is this prediction that I'm seeing consistent with other data? And that's a very broad, sensible way of validating it, but the other data can also be the position of the membrane. So if you've got two transmembrane proteins that are interacting, and the membrane region of one is up there, the membrane region of the other is down there, that doesn't make sense, right? The membrane should be in the same place, and that will usefully give you confidence in that you've got a plausible answer and rule out some obviously incorrect answers. So membrane proteins are no different really. That it's just that the membrane part, you know, and you will also see disordered regions going into where the membrane should be, but that's just you know, where you bring your expertise and sort of analysis after the fact. You look at your model critically and say, does it make sense given where the membrane should be? So it can actually be useful. It's a limitation, but it can be useful in filtering results.
Francisca Lazzaro is asking, how does AlphaFold 3 handle conformational flexibility in macromolecular complexes? Does it predict multiple energetic states or just a single most stable conformation?
Yeah, it's a great question. I mean, it broadly, I mean, one thing to say straight away is we don't, that kind of behavior has not been looked at in the same level of detail as it has with AlphaFold 2. I mentioned the idea of, playing with the MSA to make AlphaFold 2 give you greater diversity of structures. I've seen one paper where they've done the same with AlphaFold 3. There's no reason to think that those approaches won't work. So, but generally, you can be in the same position as you were with AlphaFold 2, that you've got a protein or a complex which should have a range of conformations. It can be RT state, it can be in out on a transporter, and the modeling will stubbornly only give you one of those. You can still get stuck in that position, and then you have to try and play with the MSA, include templates of the conformation that you want, basically work a bit harder to push it to produce the conformation that you want. But yeah, I think you will be able to induce it to sample alternative conformations in the same way as you can with AlphaFold 2, but it hasn't really been explored to the same extent. And if you're doing that now, you might start with looking at bioemu, which is designed for that purpose. People, playing with AlphaFold 2 and 3 to sample more conformations, that's kind of clever, but it's not what it was fundamentally designed for. Whereas bioemu has been designed from the beginning to sample conformations in the same way as you would get out of an MD trajectory. And so I don't know how it performs on complexes, but for single proteins, you might start with that if conformational sampling is what you're after.
Just got a couple more questions, which I think we have time for. Philip Robinson is asking, AlphaFold 3 doesn't allow you to input rare amino acids such as selenocysteine. Do you know of any workaround for this, or have you heard that there's any talk of addressing this?
That's a great question. No, short answer, no. I haven't really thought about that at all. I mean, you can add atoms. I don't know if you can subtract atoms and then add other atoms, so you can add atoms, PTMs and so on, but that's a good question. No, I don't, not heard of anything like that. The same, sometimes you wonder about D amino acids. Occasionally you want to model them, but no, I don't know of a way to do that.
Already answered the final question, so that's it. Perfectly on time.
Oh, right, okay.
Thank you very well. I just have a couple of housekeeping things to say, but otherwise, one of which is to thank everyone for attending. I believe we were told at the beginning that the Dan's AlphaFold 2 seminar was the highest attended Biochemical Society webinar that there'd been, and I believe this one has beaten it, because we were told that 150 was the number of attendees, and I believe I recorded 204 as at the height I saw of this, so that's good, nice to know. And people can continue the conversation online by following at Biochemical Society on either Blue Sky or X. And again, just to reiterate that Society welcomes suggestions for future topics and speakers for this Biochemistry Focus series. So if you've got an idea for a webinar for 2026, please submit a proposal, and you can find out how to do that on the Biochemical Society website, biochemistry.org. So all upcoming seminars are listed on the Society's website. And also, if you've missed any seminars in the series, you can watch them on the Society's YouTube channel. Details of that should be on screen. And also, the recording of today's seminar should be available within the next couple of weeks. Another thing is that the Biochemical Society 2027 awards are open for nominations. And the deadline for initial nominations is the 3rd of November, 2025. So if you can think of anybody who you would like to nominate, please do. Submissions are welcomed from both members and non-members. You don't have to be a member of the Society to nominate someone. again, you can visit the website to find out more about that. It's really important to remain engaged, to be connected and engaged with fellow biomolecular bioscientists. And to help with this, I'd really encourage you to join the Biochemical Society's community of researchers. And that a membership of the Society allows you to take advantage not only of that community, but also benefits like discounted registration fees for Society courses and meetings, and a wide range of grants and bursaries. So personally, I've been a member of the Biochemical Society since I started my PhD, so longer than I care to admit. And this summer, I got some funding from the Eric Reed Fund for Methodology, which is provided by the Biochemical Society, that funded a vacation student to work in my lab. And just this very morning, I finished the online R101 course that's provided by the Biochemical Society and was funded by the Biochemical Society to do that. So it really is, you can get a lot of benefits out of membership of the Biochemical Society. So if you're not a member, think about becoming a member. And that's it. We'll just thank Dan again. It was a great seminar. I really enjoyed it. And thanks to everyone, and goodbye.